Opportunity to present my PhD work. So part of my PhD work. If you want to know about the whole story, I'm defending my PhD in a month and it will be virtual, so you can all attend quite easily. So I conducted this work at the Pasteur Institute and at the Collège de France under the supervision of Roman Kozul and Olivier Spelly. And part of the work that I've done showed that genomic contacts reveal the control of sister chromosome decatenation in E. coli. So as you know, DNA is a polymer that needs to be folded to fit within the volume of the cell. And in prokaryotes, such as E. coli, which is the model that I use during my PhD, this compaction is mediated by supercoiling, DNA binding, and molecular crowding. The associated protein with the DNA form a macromolecular complex, and the space occupied by this macromolecular complex is called the nucleoid. So you have local structuring like plectonemes or chromosomal interaction domains or seats. And you also have, on top of this local structuring, a higher order of structure in the form of macrodomain. E. coli possesses 4 macrodomains, the ORI one, the TER, the left, and the right, and also 2 non-structure regions, the non-structure left and the non-structure right. The particularity of bacterial chromosome is that they're folded in both free and constrained supercoiled. The DNA is mostly negatively supercoiled, and this organization is dynamic, modulated, and modified by cellular processes like replication, or as we saw yesterday in Raphael's talk, by transcription. Changes in the supercoil can be fatal for the cell, as it can induce topological stresses, and they need to be resolved for the cell cycle to continue. So the maintenance of this supercoiling is done by a family of proteins called topoisomerases. E. coli possesses 4 topoisomerases, Topo I and DNA gyrase that Raphael talked about yesterday, and Topo III and Topoisomerase IV. In this talk, I will mostly talk about Topoisomerase IV, which is the main E. coli decatenase. It's a type 2A topoisomerase, which means it uses double-stranded DNA as a substrate. It's an heterotetramer composed of 2 subunits, PAR-E and PAR-C, and it's the equivalent of Topo II in eukaryotes. It's able to relax positive supercoiling, and it's important to know that the deletion of the 2 genes coding for this protein is lethal. So we use thermosensitive version of those subunits. So we have PAR-E-TS or PAR-C-TS. And we shift the culture to a non-permissive temperature, so here it's 42 degrees, to inactivate the Topo IV and mimic a Topo IV mutant. This protein is essential for chromosome segregation, and it will act on 2 topological stress. The first one is induced by replication. So the passage of the DNA polymerase will induce positive supercoiling ahead of the replication fork. I just realized you can't see my pointer up. Okay, so you will have positive supercoiling ahead of the replication fork, and this positive supercoiling can diffuse behind the replication forks, which will induce the sister chromatid to intertwine, which is called precatenanes. This topological stress is resolved by Topo IV, which will untangle the sister chromatids, allowing for proper segregation. But if some precatenanes remain in the chromosome at the end of replication, it would lead to the 2 fully replicated chromosomes to be topology linked together or catenated. In that case, Topo IV will interact with another protein complex called XRCD at a very special site in the third domain called diff. It will decatenate and separate the 2 fully replicated chromosomes so that the rest of the cell cycle can proceed. So this protein is very important. And if you alter it, you have bad consequences for the cell. So I'm not going to go in details in the technique, but several studies have used recombination assay as a readout for sister chromatid interaction. So they measure the level of recombination between LOXP sites. And obviously, the closer the sister chromatids are, the higher the recombination level is. And when you have precatenanes, the sisters are very, very close. So you have a high level of recombination. And that's what you see here. So you can see that in a PAR-ETS mutant, the level of recombination is higher compared to a mutant, which means that you have more sister chromosome interaction and more catenanes. In addition, if you look at the morphology, by microscopy, you can see that the cells are quite different from a wild type. Already at permissive temperature, PAR-ETS cells display aberrant morphology, so you have elongated cells. And when you shift at non-permissive temperature, those effects are even more drastic. You have very long cells and even anucleated cells. So the nucleoid is marked in green. And you can also see that in the wild type, the nucleoid occupied about 3/4 of the cell. And in the PAR-ETS, the nucleoid seems to be more condensed and centered in the cell, which reflects segregation default. So the whole point of my PhD was to investigate those changes and to see if the effect that topoisomerases, the alteration of topoisomerase IV has on precatenanes and on morphology also affect a more global chromosome organization. So to investigate the global chromosome organization, I use a technique that you all know too well, so I'm not going to present it again in this talk. So thank you for all the other speakers who presented it. It makes my life a little bit easier. So just as a reminder, it provides the genetic and spatial information at high level resolution. So especially in my case, I use the high resolution, Hi-C that Charlie presented to you today that allows us to see structure at a 1 kb resolution. So really allows in-depth analysis of the effect of several protein organizing the chromosome. In the maps that I'm going to show you, you will find features of interest above and also color-coded microdomains. So the one that we are particularly interested in today is the TUR, which is orange or red, depending on your screen. And so overall, the Hi-C reflects the average 3D organization of the genome of a population of cell. And we use that to investigate the effect of the alteration of Topo IV on chromosome organization. So this map is symmetrical. That's why I'm only showing you half of them here. So on the bottom, you have the wild type half of the matrix. And on the top, you have the half matrix of the PAR-ETS at non-permissive temperature. You can already decipher some changes between those two organizations, but to see it more clearly, I'm showing you the ratio of the matrix. So a decrease of contact in the mutant compared to the wild type appears in blue, and an increase appears in red, and no change are in white. So you can see that it drastically changes the chromosome organization, and I'm going to go step by step. So the first effect that we see is an increase of mid-range contact from the OREC to 1.35 megabase and from the OREC to 1.8. The second one are those very specific spike that emerge from the diff site and spend on each side of the diff site, on about 300 kb. So in this region, it shows that the loci that are in the TUR microdomain seem to interact with very large number of distant loci. We call these two specific signal butterfly wings, and this is the first time that we show that we've seen those types of pattern in Hi-C. The first signal that we see is at the diff region, where you have a decrease in mid-range contact compared to the wild type and a loss of the macrodomain organization. Sorry, my slides are shifting themselves. So when we inhibited replication in a way that cells either have one chromosome or two separated chromosomes, we end up losing the Hi-C pattern that we obtain in alteration of Topo IV. So you can see here that we lose the pattern, and it's even clearer when you compare the PAR-ETS replicating compared to the non-replicating PAR-ETS. In a linear chromosome, so when you have free extremities and no circularization constraint, we also lose the PAR-ETS Hi-C patterns. So since we lose the PAR-ETS Hi-C patterns when replication is emitted or when circularization constraints are removed, we hypothesize that the Hi-C signal we detect comes from precatenanes, so from the entanglement of sister chromatin. So indeed, in a Topo IV mutant, as I said, you accumulate precatenanes, and this will be the defining signal of the butterfly wings and the increase along the diagonal. To investigate further the mechanism behind this pattern, we We did a time course high C of Topo IV inactivation. So you can see on this beautiful Christmas tree that the disorganization is progressive in the inactivation of Topo IV. So just to go step by step, 10 minutes after inactivation, you lose short-range contact along the diagonal. 20 minutes after, you lose the TER macrodomain organization and have a decrease of contact in the TER, which is the blue triangle here. Then at 30 minutes, things starts to get interesting, where you have the emergence of the butterfly wings from the TER domain, further decrease of the contact at the TER, and increase of mid-range contact along the genome. Then up to 60 minutes, you have a further extension of the butterfly wings, so they seem to grow out of the TER domain, and a further increase of mid-range contact along the genome. So this phenotype, these patterns really seems to emerge from the TER domain. So it seems to start at the TER domain. And interestingly, if you remember about that special site that I told you, which is diff, the precatenanes do not seem to diffuse past this site. So the two sides don't contact each other. So we were really interesting to know what was going on in this zone. And the TER domain is defined by the complex matS-MatP. So in this TER macrodomain, you have several matS sites that are bound by the MatP protein. And this complex is very important for the structuring of the macrodomain, but also for the positioning of the whole chromosome during cell cycle. So it's a very important complex. And we looked at the contribution of MatP in our phenotype by deleting MatP in absence of Topo IV, so a double mutant parity S-MatP. And as you can see, we do not have the butterfly wings anymore. We have a perpendicular signal perpendicular to the main diagonal. And it seems now that the precatenanes can diffuse through diff and communicate together, which translate that the TER domain can now interact with the rest of the chromosome. We looked at the other part of the complex, so matS. So we looked at it by displacing some of them. We did so by using a strain that has an inversion between 0.8 and 1.4. And as you can see, we moved some of the matS sides up. And now it has, as a consequence, the first butterfly is enlarged, and the new border of the butterfly coincides with the last matS site that is moved up. Interestingly, you can also see that now we have a matS site gap, where it seems to coincide with a sort of diffuse signal here. So we use those results to try to create a model. So if you look at what's going on in the TER macrodomain and the diff are here, you have an accumulation of precatenanes because of the absence of Topo IV. And in the absence of MatP or in the absence of matS sites, you end up with the precatenanes that are able to cross past diff and interact together, which would mean that in presence of matS and MatP, you have this complex that is blocking the precatenanes from crossing to the other side. So we define this complex as a decatenation hub, which would mean that in the wild type, the precatenanes are drawn to this decatenation hub to be resolved before the end of the cell cycle. But in the absence of Topo IV, no one's here to decatenate, to resolve those precatenanes. So they will accumulate here, and this will result in the disorganization of the nucleoid that I showed you with the butterfly wings. Another key determinant of the TER macrodomain is the E. coli SMC MUGB. So interestingly, MUGB has been shown to interact with Topo IV and stimulate segregation. And also MatP in the TER domain inhibits MUGB activity in wild type cells. So basically MUGB is kept out of the TER domain by MatP. And recently, Vicky Liol and Axel Kornak showed in 2018 that MUGB promotes DNA loop formation. So it seems to be a cool candidate to look at in our pattern. So we deleted MUGB in absence of a functional Topo IV. And you can see that the shape of the butterfly ring drastically changed. They seem to be shorter. So we integrated MatP in our model by saying that MatP could promote DNA loop formation and thereby sort of separate the sister, which would give maybe more freedom for the sisters and the precatenanes to communicate with each other, hence the length of the butterfly wings. So in conclusion, we said we hypothesized that the combination of matS-MatP and Topo IV creates this decatenation hub, and the alteration of Topo IV results in a cripple decatenation hub. So as a take-home message, sorry again, we propose that Hi-C is now able to give a new readout of topological constraints, in our case precatenanes, that the absence of Topo IV gives rise to an accumulation of unresolved precatenanes that accumulates at the border of the term macrodomain and are not able to contact the rest of the chromosome, which we call butterfly wings. The position of these wings are dependent on the term macrodomain organizers matS and MatP. The length is dependent on the E. coli SMC MatP, and the alteration of Topo IV results in a crippled decatenation hub. So I would like to thank my two labs, Roman and Olivier, for supervising me on this project, Charlie for also supervising me on this project, and former members of the Espeli lab, Hafez El Sayed and Ingrid Bio, who worked previously on this project. As I told you, my PhD defense is very soon, so if you're interested about this story, just tell me and I will gladly invite you to the defense. Thank you very much, and I would be happy to take questions.
Jean-Marc, ton micro.
I felt really alone.
Sorry. Thank you, Brenna. So I kept my microphone out. So really sorry. I'm looking for questions. And Cedric, are you there? No, I think he had to go. So I have a question. Do you think that this mechanism is very general for bacteria in general?
Okay, so this is a very cool question, and we're working on that at the moment. So I'm doing the same experiment in Salmonella, which is pretty cool because it's a pathogen. So we could see if the mechanism is conserved. I think it could, but it's true that it's something we have to look into. Because, for example, MatP other bacteria has other SMCs, like Bacillus has other type of SMCs that do different things. So it could change the mechanism a little bit.
Okay, sure. So Aurel.
Hi,
hey,
Aurel.
Hey, how are you? It's very interesting. The butterfly is independent on the MatPF. I mean, yeah. it is shortened, but it still go up to 1 megabase, which is a lot.
Yes.
And if you imagine that this is a sort of a sliding of the two sisters against one another, then I wonder what drives this sliding that is so extensive.
Yeah. So it's true that it was surprising that the MatP didn't erase the butterfly. Like I was expecting more of a drastic effect. I don't know what drives this sliding. I don't know if it's a constraint, like physical constraint. I'm not good at physics, and I'm not a physicist, so I can't really do that. Maybe we need a model to see what kind of strength will happen, because obviously I think the other proteins that are trying to segregate might pull somewhere to do this sliding, but honestly, I don't really know.
Okay.
But that's a good question. Thanks, Aurel.
Okay. Vicky, tu veux?
Yes. Hi. I don't know if you listen to me now.
Vicky.
Hi, a very nice talk, Brenna. I
thanks.
really like it, and I would like to be invited for your presentation.
Yeah, sure.
Well, apart from that, the question is that first that you are always showing ratio maps.
Yes.
So, and I see that in fact this is not a really strong effect. You are like no more than 2 times. That's one question. So how you are sure that this is a direct effect? Because in fact you are analyzing things which are in fact a homologous DNA, and we cannot see that by Hi-C. So yes. How do you imagine all this? No?
Yes, so exactly. So first of all, I think maybe it's a bias of Hi-C that allows to visualize short to mid-range contact better than long-range contact. It's true that I was having difficulty really observing because it's a very, very long-range contact. I also think the fact that it's not very strong might be because it's sporadic in the population. So you might not always have the same length of the butterfly in each cell, which gives kind of this diffuse, not very strong signal. But if I can share my screen again, I can show you, for example, in the other subunits of the Topo IV. So in part C, we have a much stronger, can you see my screen?
Yes.
Yes, okay. So I'm sorry, I'm taking 5 seconds to do
it's okay.
that. Oh, come on. Okay. So here you can see it's much stronger. In the part C mutant, the signal is way stronger, and we also have a double mutant combined with Topo III that shows an increased signal as well. And to justify the fact that it's precatenanes, indeed, you can't visualize sister-to-sister loci contact in IC. As you said, they will match on the same position. This is why we envision this sliding catenanes thing. So instead of being right in front of each other, so they will be slightly separated, slightly shifted, so a little bit slided, which will mean that you can have contact between the sisters, but not directly between like locus A and locus A prime, but more like A and B primes, stuff like that. So it's really because of this sliding that we think we can detect inter-sister chromosome contact. It's true that we have strong hints about the fact that it's precatenane, like it's dependent on replication, it's dependent on topological constraint of the circular chromosome. But it would be cool that one day we can see the precatenanes, which I know is a big thing to try to look at. So
yeah, I know there are some techniques you are developing on that, so it's okay. But the question is also in the sense that if you look at the weird events, because when you analyze Hi-C contacts, at least with 3C, we get a lot of what we call weird events, which are the one read exactly the same than the other read, and these are basically what you are looking at. So did you see an increase in these events in your?
No. So I have the same amount of events are as in a wild type, like, you know, the pi that we obtain. I don't know if we use the same
yes.
pi, but my 2 pi's looks the same. Yeah, but it's something that you're right. I will look at it. I hope I'm not analyzing noise or anything, because then my whole PhD falls apart. But yeah, it's something that I will look at more deeply. Yeah.
Okay, thank you very much.
Thank you.
Alors, on a une question, Vittore.
Ah, Vittore!
Hi. Very nice talk. I was very puzzled by the time course. That I think is very convincing that the effect is true and is progressive. I wonder if you could imagine, or you did, a dependence of this time course from the growth rate, like if this will change something, maybe.
So I didn't look at it. You mean like the timing of replication would change the effect?
The velocity of the effect, maybe, and
yeah. Okay. So we could look at that in more deeply as well. I think that we didn't. So it's not synchronized cells as well. So it's true that they're not all growing at the same time. So I don't know if we synchronize them. It's a little bit hard to do since I'm already using a thermosensitive system to synchronize. I mean, it's a technical mess, but we should try to do it. But yeah, the timing, if we synchronize them, maybe will be even like more striking. I don't. But yeah, I didn't.
And another question is, you could maybe try to do the same in the reverse, like
yes, that would be really cool. So I did the complementation where I expressed the functional pari subunits in my pari TS, and we restore completely the phenotype. It's not inducible, so, but I could try to do that. Another thing that I did is that I induced MatP in the MatP pari TS to show that you go from the perpendicular signal to the butterfly wings again. So I do have a sort of synetic like that, where you reinstore the decatenation hub, or you reinstore the butterflies. Yeah, I could have shown that actually. But if you want to see it, you can come to my thesis.
Sure. Thank you.
Thank you, Vito.
Ok, je crois qu'on a fini avec les questions. Merci déjà à tous les orateurs de la dernière session de la matinée.